Hi everyone, my name is Lauren Buckhout, and today I will be presenting my final genetics portfolio project on the disorder hemophilia A. Hemophilia A is an X-linked inherited disorder and affected individuals lack the clotting factor 8, which inhibits blood from clotting properly. The graphic on the right side of the screen is a visual representation of how hemophilia affects clotting compared to the normal clotting process. The blood vessel on the top is a normal blood vessel with a tear with clotting factors present, working to repair the vessel like normal. And the vessel on the bottom shows a vessel belonging to a person with hemophilia and uncontrolled bleeding at the tear site due to the absence of the clotting factor. So people who suffer from hemophilia and abnormal clotting ability experience a range of symptoms which can vary based on the severity of their disorder. But affected individuals may experience excessive bruising, bleeding under the skin, hematomas, bleeding into joints, and spontaneous bleeds and brain bleeds, which can be life-threatening. Women who are carriers of the hemophilia gene may not have normal clotting factors and experience symptoms like heavy menstrual periods, susceptibility to bruising, and excessive bleeding after childbirth. As I mentioned before, people with hemophilia A specifically lack the clotting factor 8, which is located on the long arm of the X chromosome. Factor 8 is a plasma glycoprotein, as we know, has an essential role in blood coagulation. The disorder is caused by gene variants in factor 8, and the genomic location of F8 is XQ28. Hemophilia A is inherited in an X-linked recessive pattern. We know that males inherit their X chromosome from their mother, and females inherit an X chromosome from each parent. The X-linked recessive pattern mode of inheritance provides an explanation as to why males are more likely to be affected by hemophilia A. Females who are carriers have the ability to pass on the gene to male and female offspring, and affected males have the ability to pass on the gene to female offspring if there's a cross between an affected male and an unaffected female. And that's shown in this visual here on the right side of the screen. Here's an example of a pedigree that tracks hemophilia A within a family. The squares represent males and the circles represent females, and the key on the left side shows how the pattern or shading of each shape correlates to how the disease is expressed. So you can see how the family started with an unaffected male, Daniel, and a carrier mother, Claudia, and they produced together a carrier daughter, an unaffected male, an affected male, and an unaffected daughter. And those are all the possibilities of their cross. But as the pedigree expands, you can see that there are more possibilities for crosses. For example, Harry and Kelly. Harry is an affected male and Kelly is an unaffected female. They produced an unaffected male son, and due to the nature of the X-linked recessive inheritance pattern, their cross could result in either the unaffected male that they had or a carrier female. So here's some historical facts about hemophilia. Some of the earliest recorded incidences of excessive bleeding date back to the 2nd century AD. And those were among Jewish writings documenting excessive bleeding of baby males who had been circumcised. In the early 1800s, scientists began to recognize a hemorrhagic bleeding disorder that mostly affected men in certain families. And then in 1828, the condition was recognized as hemophilia, although it was spelled differently. In the 1900s, advancements and research identify the disorder and the mechanism to be connected to the absence of clotting factors specifically. And sometimes it was referred to as the royal disease because it affected the royal families of England, Germany, Russia, and Spain in the 19th and 20th centuries. So according to the CDC, hemophilia A affects 1 in 5,000 male births, and about 400 babies are born with hemophilia A each year. While the exact number of people living with hemophilia A in the United States is not known, the CDC tracked during the period of 2012 to 2018 as many as 33,000 males in the United States who were living with the disorder. So for each man with hemophilia, 1.6 female carriers are estimated to be identified. Rarely, a woman can inherit an affected X chromosome from both parents. So that would be from a carrier mother and an affected father, leading to full expression of the hemophilia variant severity. In the study that I cite here by Van Galen et al., they determined that hemophilia carriers may not receive full care of hemophilia treatment or comprehensive care in comprehensive care centers, and therefore are likely underrepresented in surveillance databases, limiting researchers' ability to characterize their symptoms and diagnostic journey. So in terms of testing, it's possible to detect hemophilia in unborn fetuses by sampling or percutaneous umbilical blood samplings. Some screening tests, such as the complete blood count CBC, activated partial thromboplastin time APTT tests, and prothrombin time tests, and fibrinogen tests can give doctors an idea of how well or not a person's blood is clotting. And then there are other tests, molecular and carrier screening tests, that can give more information about the severity, possibly, of the disorder, and those can include inversion analysis, screening analysis, and deletion duplication analysis testing. So early detection of hemophilia A is important to create the best treatment and management plan for an individual. And while there's no cure for hemophilia, depending on the nature of an individual's diagnosis, there are some treatments available. For example, the administration of the specific clotting factor that is lacking in hemophilia A's case, that would be factor 8. And that can be life-saving in some circumstances. And that is all I have for you today. Here's a page that lists my references that I used in this presentation. Thank you.